Hello, I'm your host, Ray Dogum, and welcome to VibeCast. Thank you for joining us as we explore the exciting advancements in technology-enabled collaboration to excel in important drug development. VibeBio seeks to find every cure for every community. We think big as no one should be left behind in the pursuit of living a healthy, happy, and productive life free from disease. Collectively, we have the skills, we have the technology, and we have the passion. We now need the community catalyst to bring that all together. That's Vibe. We see a future where communities of biopharma experts and patients collaborate to identify high potential medicines and have the ability to access capital on demand to actually develop them. The VibeCast is our weekly informational podcast where we explore some of the hottest topics in drug development and technology innovation with some of the dynamic people that make up the Vibe community. Join us to learn, imagine, question, and help us identify and develop solutions together. Our guest today is Dr. Mark Kotter. Mark is a stem cell biologist and neurosurgeon at the University of Cambridge. He is founder and CEO of bit.bio, as well as co-founder of a cultured meat startup called Meatable. Mark, welcome to the show. Really appreciate you being on here today.
It's a pleasure to be here. Thank you for the invitation, Ray.
Awesome. Maybe we can get started on where you are today.
Right. I'm right there in the headquarters of bit.bio. We're located at the southern fringe of Cambridge. That's an old university town. We're talking about Cambridge, UK. And it's a very vibrant biotech community here. It's the largest in Europe.
Excellent. And let's maybe tell the audience a little bit more about your background. So maybe, share some insights into how you got to where you are now.
Sure. So I joined the University of Cambridge first for my postgraduate degrees, but then again as junior faculty, naively thinking I'm going to create cell therapies for spinal cord injury patients. So that was my ambition and my mission. I'm a trained complex spine neurosurgeon, and the main patient population I look after are spinal cord injury patients, traumatic and non-traumatic. Obviously, when you start out on a journey, you don't know how complex this field is and how complex, in particular, cell therapies are. But what I did know and understand is that very complex conditions need different classes of drugs. If you think about small molecules, biologics, essentially these are dead molecules. And the difference to cell therapy is that the cell therapy can replace lost cells, which of course these molecules cannot, but also cells are flexible. They can react to the environment. So you could call them a class of intelligent drugs. And of course, we've seen the first evidence of their superpower in the field of CAR T cell therapy.
Yes, and we'll dive into CAR T cells hopefully a little later as well, just a bit. How does your experience as a spinal cord surgeon help inform your biotech business strategy?
Well, I think the first thing is, of course, you're absolutely focused on patients. It is really my choice in life to become a doctor. It's a huge privilege to be in front of patients. I derive incredible, let's say, inspiration from them. My particular patient population, often struggles with breathtaking changes to from one day to the next. And I think seeing that there is really nothing else but surgery that we can do for them, and the limitations of that, has really driven me to become very interested in regenerative medicine, start do my postdoctoral training, start a lab in this field. The other thing that I think is really important is beyond the particular one thing that I'm focused on. There are so many other needs that patients might have that are not recognized. In fact, one of the biggest things is this disconnection between what patients require and what health professionals and pharma companies, biotechs think that they require. So to address this, I also spun up or created a charity. It's the largest charity in a particular area of spinal cord injury, with more than 4,000 patient members now. And the dialogue is and has taught me so much. So I'm a very passionate advocate for patient-initiated research and patient-initiated, activity, all the way to translation.
That's a great point. And I think patient communities have, especially in recent years, maybe in the last decade or so, have really gained their voice, or their voices have become louder because of the social media platforms, because of just better engagement with providers. So I think that's really great. And let's talk a little bit about your charity. I'm just curious. Can you share a bit more information of when you started it, or when you got it? Yeah.
I mean, rightly so. There is a disconnect between health professionals and those that actually experience disease. So this started off as a way of engaging with a group of distributed patients on the internet to get input on a research program that I wanted to submit to one of the funding bodies in the UK. And
is that a requirement for the UK?
It wasn't a requirement.
It should be, right? But yes,
it's now becoming more and more a requirement. But at that point in time, it was something that was not required. But you could, if you could demonstrate that your plans are aligned with patient needs, obviously that's a benefit. So I tried to get some feedback on a trial that we wanted to conduct, which is called recede myelopathy. So the condition is cervical degenerative myelopathy, which is caused by wear and tear changes in the neck that compress the spinal cord and then lead to a slow motion spinal cord injury. And so I was able to use the internet to approach many more patients in a very short period of time and get really diverse feedback on our plans. And this sort of crystallized, activity online. And we then found another individual that actually suffered from this condition, who also gathered a group online together. His name was Owen Sadler. And we came together, we met, and we sort of created we called it the first Cambridge myelopathy away day. So we invited a number of patients, brought them into a room. And it was one of the most emotional experiences that I actually ever witnessed. These patients are very isolated. They don't know about the condition. Meeting others with the same condition, was incredibly emotional. But also we saw that the power of getting to them together, the shared experience and the shared knowledge and the shared frustration and the shared expectations and worries. And so we decided to create over, a online group. And from that online group, we then decided just to incorporate a charity. And now it is still small, but in terms of patient numbers, we are 4,000 plus. And it's an opportunity for others to meet others with this condition. They get trusted information at the charity is called myelopathy.org, and we've been able, together in partnership with the charity, to really redefine the field. We conducted what's called a research priority exercise, which was supported by the James Alliance. So there's a very structured process that identifies patient needs and formulates research questions. We conducted a definition of a core outcome set, a core data element set, in order to help standardize the way that you record clinical trials and conduct clinical trials. So quite transformational, quite frankly, incredible experience.
Yeah, no, that sounds amazing. And I'm glad that you started it. And it sounds like, the community is growing in there. So I think hopefully after this VibeCast, more will join, or at least understand what you're working on. So be more familiar with it.
Thank you.
Let's switch gears and start talking about the overview of cell therapy. And just the entire landscape in the marketplace now and how that's evolved over the last decade or so.
Yeah, I would say the notion of a cell therapy, and that might be something exciting, predates what we have right now in the clinic, which are mainly CAR T cell products that are focused on oncology. Remember, 10, 15 years ago, everyone talked about regenerative medicine and how stem cells would provide a source of cells that we could use to treat all sorts of different conditions. Turns out that this is very complex. And it turns out that there were some unexpected quick wins on the oncology side. So this started, of course, with what's now called CAR T cell therapies. Those therapies are donor-derived, so patient-derived material. So you basically you take blood, you filter out the T cells, which are a kind of white blood cell, and then you engineer them with a CAR, which is a receptor that recognizes cancer cells. And when you infuse them back into the patient, what we learned is that you can cure blood malignancies, so blood cancers, at the latest stage where actually patients had no other hope. So 50% of patients that have this particular condition and underwent all the sort of regular treatments that are available and would have ended up with no hope whatsoever can be cured by a single treatment. Mind-boggling. It's mind-boggling. To me, this is the rather white flight of cell therapy. It's really a very poorly, very simple, let's say, engineered cell. A little bit like, the aeroplane that was built that was stuck together nearly by sellotape. But it lifted off, and cell therapy lifted off, and has created this incredible, paradigm. And of course, that led to company creation. That led to an interest of pharma. So it's a phase shift that has occurred based on those discoveries.
And you were mentioning how these patients who had blood cancer might have gone through regular treatments without success, and then the CAR T was able to essentially cure them. And oftentimes there are many medicines that take years, decades, at least a decade potentially, to go through the entire process through drug discovery and preclinical and clinical trials. Is that frustrating to you, that the time it takes to get to one point and to, from bench to bedside as a physician, right? And do you believe that there is a path, a way to develop our technologies or data or even like wet lab science more quickly to deliver drugs or to deliver treatments really to patients more efficiently?
I think there's a number of things that you can do. So let's step aside of cell therapies and look at the entire drug discovery process. We know the conditions that we've been trying to treat for a long period of time, and where we were quite unsuccessful or struggled to develop drugs. I mean, neurodegenerative conditions, for example, is very hard to treat class of disease. And so in order to treat those or find therapies that can treat those conditions, pharma or biotech companies often start with a concept and then an animal model that they use to develop their drug. And the problem, of course, is that whilst there is some similarity between human and animal models, it seems to be sort of at greater distance. There's a greater translation gap when it comes to the actual disease. I'll give an example. There's not a single mouse on this planet that has Alzheimer's. So when scientists try to develop a drug for Alzheimer's, they take a mouse, they modify the mouse so that it gets a disease that looks like Alzheimer's. Then they do a very efficient drug discovery process. And when we test the drug in patients, we figure out that the thing that is in the mouse is a different disease than Alzheimer's itself. So the first thing you need to do is you need to start with human cells that actually have the condition, and that will probably double, maybe multiple the chances of success, which means fewer clinical trials with higher probability. But if you only would double the chances, you would reduce the cost. If you include the failure rates, we spend about 2 billion on a drug, by about half. So there is one big chunk of efficiency just by sort of starting with the right material when you develop your drugs. There are other nodes where I think we can be more effective when it comes to clinical trials. It's quite mind-boggling, but what we do is we take minimal measurements across a large set of patients. So from a stats point of view, that gives you your p-values, etc. But it's a little bit unintuitive because you could flip it on its head and you could say, let's take lots of measurements on a few individuals. And I think so if you use sort of new modalities, new biomarkers that you can cover sort of maybe even 24/7, that would allow you to massively reduce the number of patients that you need in a trial, which again could be a step change when it comes to drug discovery. And that is needed. We need to be efficient at the preclinical stage and at the clinical stage, because science is exponentially increasing in terms of our knowledge base and understanding. But if we have such a tight, let's say, eye of a needle that we need to thread our drugs through, will not be effective. So we need to really rethink the paradigm and fine tune every aspect of it.
Yeah, no, absolutely. I think also if we're collecting more data from less patients, the issue will become that there's a lot of variability in the data. So I think getting to the point of approval may be more challenging, but I see what you're saying too, where
I think this is a mathematical thing, to be honest.
You can optimize.
So, I mean, if you especially these chronic degenerative conditions, if you just take a reading before, the trial and after the trial, that's not helpful. But if you have longitudinal data that demonstrates, change over time, you're in a very different mathematical construct, and that allows you to, be a lot more efficient.
Yeah, and if you want to even increase that p-value even further, we can have lots of patients with lots of data, and then we'll be able to essentially. But again, there's risks with clinical trials, and so I totally see your perspective there. How is cell therapy different from gene therapy? Can you sort of suss that out a little bit for us?
Sure. Yeah. So cell therapy is really the treatment becomes a cell. And we've discussed already, how this is very different from the small molecules or the biologics, because you're actually infusing in the bloodstream, for example, or transplanting cells into a part of the body. So we've done this successfully in the CAR T space. There's been experimental treatments to treat Parkinson's, where dopaminergic neurons were injected into the brain itself. There's been attempts to repair spinal cord injury with cells. So that is the concept of cell therapy. Now, gene therapy is quite different. Essentially, what you do is you take a virus that contains genetic information, and that virus is then again administered. Most of the virus are based, or most of the gene therapies are administered via the bloodstream, and this virus or these particles then infect the cells that you want to treat or correct. There's two issues, of course. First of all, you don't have much control where these viruses go, and they have a tendency to infect a lot of different cell types. And of course, if you think about that, having a new virus and lots of different cell types can be a challenge. So we've seen, for example, a classical disease where this approach could be transformational is a monogenic condition like, let's say, Duchenne. But the problem is, how do we make sure that the virus only really goes to the muscles and doesn't infect all the other cell types that we have in our bodies? So can we channel that virus or that construct particular to a muscle? And then if we can't, is there a second mechanisms where we can ensure that the virus only gets activated in a muscle? So these are the two tools that we have. And I think there needs to be a lot more research done. We've been good at treating some monogenic diseases with gene therapy, especially if they're not involving that much bulk of cells. Duchenne is an extreme example. We've got a lot of muscle bulk in our bodies. So if you want to correct that, you probably need a high volume of these viruses. And then you have a lot of those going at places where you don't want them. But if you have something that's a bit more localized, maybe, and you've got a better way of targeting those constructs, then you could really create some transformational treatments, which are already available. So this is not science fiction. Again, I think we've made good progress there.
That's what's so exciting about this field. It's growing so quickly, and I think the tools that biotechnologists have now are just so powerful. And we've only beginning to embark on this journey. I
agree.
In human society, it's really it's
overwhelming
sometimes, but it's really great.
I think it's mind-boggling. It's wonderful how we see this. Our capabilities grow, and we get a much more comprehensive understanding using our ability to sort of essentially read the entire genome, sequencing that entire genome. And what I'm really excited about is the tech stack that we now could sort of label as synthetic biology. So that is sort of, this is, going away from, a simple molecule to something that is a very sophisticated toolbox that allows you really to engineer biology. And we're using this for cell therapies. CAR Ts are engineered. We can use this to create better disease models, again, to accelerate drug discovery. But I think we can also use it to create transformational cell therapies and make them affordable and available. Because at the moment, and that's one of the big bottlenecks of cell therapies, they're incredibly expensive, $500,000, $600,000 in the US. But there's only one cell therapy that I know of that is actually turning a profit. All the other ones are loss-making. So that's not a good business model. But it also restricts the use of these therapies, because if you have to subsidize this, and if the costs stay that high, then you can't target something that is not deemed as being high resource intensive as oncology. So we really need to bring those costs down.
Yeah, you make a great point. Like the funding for getting the treatments that are approved to patients is so expensive, or it's so difficult to find or expensive. So we have a lot of funding gaps in the whole process, including basic research all the way down to getting them into patients directly.
But it's not only funding gaps. It's also, if you think about what capitalism should do, is make things better and cheaper. That's driven obviously by technology. And I think we need to use the new technology and entice the capital to make things more affordable and more prevalent. So I think that's the principle.
And I think that will happen over time. I think we're in a state now where we're trying to figure it all out because it's all happening really quickly.
It's all new.
Yeah, it's all new. If you think about insulin, right, when it was created using animals, it was rather expensive. I think it's gotten much cheaper as we started using synthetic insulin, right? So very curious to see how synthetic biology will impact the cell therapy space. It's almost an infinite potential there. So it's really, really cool.
I agree. Any disease that is severe enough probably could be treated by a cell therapy.
So let's dive into bit.bio and what is the critical problem bit.bio aims to solve with accessible cell therapy technology?
So it's really what I just mentioned earlier on. Cell therapies at the moment are too expensive and the products are too variable. So the question is, and they are also sort of just at the moment restricted to immune oncology. So the question is, can we find another paradigm that allows us to manufacture cells with extremely high consistency, the consistency that you would expect from a drug, and at scale, and make cells accessible for not only patients that suffer from oncology, but also other conditions. And so bit.bio has industrialized the manufacture of stem cell derived cells. And by this, I mean any cell. So we have an end-to-end platform where we can, recreate any human cell type in theory, but we validated the entire discovery platform from concept to cell type, and then create very scalable, very consistent manufacturing paradigms that allow us to make those cells really accessible. So the proof is in the pudding, obviously, but you can see bit.bio not only creates cells for cell therapy, which we develop either alone or in partnerships towards a clinical trial, but we've also released what we call research products, so IO cells. And those are cells that scientists can actually now use and validate and implement in their drug discovery processes. And the feedback we're getting is really very positive. So these are very, very good cells that very much represent what we have in the human body, although they are created using a synthetic biology paradigm.
Any upcoming announcements or any information regarding new drugs in the pipeline that you'd like to share?
So I unfortunately, I won't be able to share or make any announcements at the moment. They come when they come. That's part of the constraints of being in a company and not in academia. But I'm very excited about the pipeline.
That's excellent. How can regulations or, changing regulations or improving regulations help to accelerate research and even accessibility for cell therapies?
So first of all, I want to shout out that some really groundbreaking concepts have been embraced by the regulators. We saw recently an announcement that by the FDA that said that you don't actually have to do animal experiments if you can show, if you can use human cell models, for example, for your product. That's a phase shift. So I'm not sure how many, drug developers will ditch animal models, but it demonstrates an openness to what the future holds, and I think that's so fabulous. The second thing I want to say is regulation is incredibly important. We are here, developing drugs that will affect, people. And disease. So we have to make sure that we can do everything to keep those things safe. So again, regulation has a track record of making sure that, and they've been flexible if you think about the new trial design. But a heavy lift is at the same time is occurring and has to occur at the moment. Regulators have decades of experience with small molecules and biologics, but we have very, very little experience with gene therapies and cell therapies. So we don't know all of the risks. We don't know which ones are really important, which ones are less important, if you think a bit broader. So at the moment, we are in the learning phase and the regulators are in the learning phase. And what I would say, one of the things that I really would like to embed in the regulation is this concept of, regulating platform technologies, maybe different. So cell therapies, for example, is such a platform. It's really a process that is established and regulated. And so you can take cells from any sort of individual, and if if that sort of is processed in the same way, the product can be released back to patients. Now, when it comes to centralized cell therapies, donor-derived cell therapies or iPSC-derived cell therapies, we sometimes see a tendency to sort of say this genetic background is a product, the next genetic background is another product. And I don't think that I think we should think broader about this. I think we should sort of maintain a process view of cell therapies. And I think something similar has occurred in the mRNA space, where initially individual mRNAs were regulated, but now there's a framework in which you can, for example, create mRNAs against particular personalized cancer indications. And so this idea of regulating an approach versus a single sort of product, I think is going to be very important if you want to leverage, the incredible opportunity of synthetic biology.
That's really great. And, I do agree regulation is important, especially in this space where we're still learning. And there needs to be that checks and balance between a capitalistic drive and, the society at large. So I totally agree with you there. So looking forward to some progress. As you mentioned, there is some already. Hopefully we'll continue that trend.
Yeah, no, I think what the regulator requires is more funding because, they have their own bottlenecks and constraints. And if we want to be serious about, expanding the portfolio of things that can be treated, again, we have to remove those bottlenecks.
That's a good point. I think prioritizing health hopefully will become really a priority, right, for society and for governments and things like that, because essentially that is what we can learn from doing that is, again, limitless. So really,
this is the most worthy moonshot. Obviously I'm biased. I'm a doctor, but I think curing certain diseases, and I think we are now at a stage where we have the tools where we can create such moonshots. It's very exciting.
And just the idea of eliminating or reducing human suffering can make the whole world a better place. And
hugely inspiring, no? It's hugely inspiring.
Yeah, it is. Can you share some advice for aspiring scientists or physicians who are interested in pursuing a career in cell therapy?
So first of all, don't be afraid. It feels like a daunting thing, and especially under current market conditions, where there was a downswing in biotech. The capital that's required for cell therapies, is sort of causing questions. But here's the paradigm. It's unavoidable. For the next sort of transformation in medicine, we need that new class of drugs, intelligent drugs. And the opportunity is incredible. If you think about what diseases we can actually treat in the old paradigm, it's very limited. But what cell therapies could treat is incredibly broad. So we've also got a lot of preclinical evidence that cells, when they are administered in disease models, ranging from CNS disease to heart to metabolic disease, liver, can address those conditions. So it's really the fundamental basis of the applicability is there. And I'm also, so that's one thing. So that there's a huge use case. But the second thing is there are techniques and methods and technologies that are being developed around the globe, but also in bit.bio, that will really drive the costs down, dramatically. So this is only a matter of time, and that's why I think you can really, this is such a huge opportunity. Another point, if you look at the IRA, the new Inflation Reduction Act, there is certainly a bias towards novel modalities because government understands the importance of actually developing these. So there's political support. And finally, if you think about the interests of big pharma, whilst, maybe VC funding at the moment is a bit more difficult to obtain, they have really, re-emphasized their interest in this space. So I think we're in a maybe environment where it's a little bit harder at the moment to start something up from scratch. But the good news is there's a very broad consensus that this is going to be transformational. And there's a real push to make this happen.
Let's dive a little deeper into the funding landscape too. So I understand bit.bio had 3 funding rounds, raised a total of $145 million. That's really great. And I think the last round was a Series B in 2021. So I'm wondering, what's bit.bio's next inflection point, or when do you think their next? So that's one question. And then when do you think their next round will be?
Yeah. So actually, we've sort of raised up to close to 200 million so far.
Thanks for correcting that.
And as a biotech, we are obviously continuously raising money. That's part of the job of a biotech. But the good news is, of course, that bit.bio also has a revenue stream that is growing now from the research product side. And that allows us to more and more substitute the platform using organically grown revenue, which I think is a very good thing to have.
That's better than raising money. I would definitely say. I would agree.
I would say so too.
Yeah, no, that's great to hear that you've reached that milestone. Of course, early biotech companies often don't have that revenue internally.
No.
So, awesome. And one other topic I wanted to just touch upon is this idea of using synthetic biology, like you mentioned, as a software approach to medicine, sort of like very granularly being able to modify cells in that way. So final question, I guess, what do you think the future will look like with the software approach to medicine?
Okay. So of course, this is another way, programmable biology, synthetic biology, software approach, really all is a way of describing a new paradigm in biology. In fact, there's a quote from Sidney Brenner, got the Nobel Prize for programmed cell death, that says, we need to rethink biology, and we need to realize that at the center of biological theory, there's a code script. And this code script is, of course, the DNA, which is the storage of all genetic programs. That's tightly packed together in the nucleus, and you could say this is the hard drive of a cell. Now, the key thing is when you turn on a gene, when you turn on information in the DNA, it translates into the physical realm of the cell, the proteins and the lipids. So in this case, software information controls hardware, which is very different to when you look at a computer. So these genes that are encoded in the DNA, they interact with each other at the protein stage and also the protein DNA level, and they create what's called gene regulatory networks. Another way of labeling this would be programs, software programs. Now, and these software programs dictate the identity of the cell and the function of the cell. These programs are controlled by a set of genes that are called transcription factors, and they act like code words. We've got 2,000 transcription factors in our genome out of 20,000 genes. And what they do, they activate or shut down other genes. And so these are the regulatory sort of elements, literally like code words. And so what scientists have demonstrated, and this started with Harold Weintraub in the 1980s and then continued to Shinya Yamanaka and Marius Bernig, is that if you activate the right transcription factors, you can actually create or turn one cell type in another cell type. So you can program cell identity, and that's really the basis of bit.bio. So we can essentially create very robust manufacturing paradigms by tapping into this programming biology approach using transcription factors. Our secret sauce isn't so secret. We use genomic safe harbor sites. That's another sort of story. This allows you to make it incredibly robust. But beyond that, the paradigm is even larger. You can not only recreate every human cell type, you can add functions to cells that exist or that you can program. And you can actually mix functions. So you can actually create cells that have traits from 2 different cell types. There's been experiments that show that you can combine a neuron, a brain cell, with a muscle cell, or an immune cell with a brain cell. And so you can, the point that I'm trying to make here is that our software is a lot richer than what we see in a human body. And what that means is there's even more potential to create something that actually can address, certain conditions. You can design a cell with functions in theory that you can then use as a novel kind of medicine. In some ways, when you look at CAR Ts, that's already what we're doing. We are basically giving a normal T cell a receptor, and that receptor recognizes cancer. That's not a natural receptor. This is a receptor that has been engineered. So what all of this means is that we have reached an inflection point in life sciences, where we now can apply a software approach, which entails 3 steps: reading, writing, and executing genetic code. Reading, of course, is sequencing. Writing, CRISPR, base pair editing, and then executing. This is not an issue at all when it comes to lower species E. coli that we've used for example, to generate insulin, but it's a very high bar when it comes to mammalian and human cells. bit.bio has their own solution for this called OptiMax. And what it allows you to do is like a control system. If you insert that into a cell, you can deterministically, i.e., you can dictate the program that's on in that particular cell type. And by switching the program, you can create all these things that we just talked about. So it's really an inflection point at the moment. This whole area we could call synthetic biology, but the tools that we now have at our fingertips are extraordinary. I mean, transformational. And I feel so privileged to be born in that period of time and be part of that, transition.
Yeah, no, I feel you, and I feel the same way. I feel very like we're in the golden years of biotech, and synthetic biology technology coupled with a better understanding of epigenetics as well, and just the environmental factors that affect cells and systems. There's just so much potential there that we are unlocking every day. So it's really exciting.
I agree. I think we have a unique opportunity to really change the trajectory of many different conditions.
So the real final question, actually, apologize, I know we're running short on time, but do you find that there are certain risks or concerns, like existential concerns for humanity, when you think about synthetic biology to that degree?
So I think when it comes to cell therapies, probably not so much. If you take a cell outside of an incubator, it just dies. Yeah. So it doesn't spread. But of course, what we learned is that the viruses can get out of hand. And of course, bad guys could develop things like that or other pathogens. So when you have, when you let people go rogue on pathogens, bad things can happen. That said, nature and the evolutionary pressures of nature, can create a lot of conditions. The recurrent, infections that we all suffer, a part of that, process. But I do think, again, regulation is important. Making sure that there's checks and balances, making sure that SynBio is regulated. That'll be an important aspect.
Dr. Mark Kotter, thank you so much for joining me today on VibeCast and chatting with us about cell therapy and the landscape and what bit.bio is up to. Any final thoughts you'd like to share with the rest of the community?
Well, I think we are both excited about what's in front of us. And thank you so much for the opportunity to be with you.
Excellent. Thank you.